So today we're going to be looking at transport into the cell and out of the cell. This is actually incredibly important as we talk about our system. So we're going to be talking about the digestive system and how stuff gets into blood and out of blood. And we are going to be talking about the respiratory system and how stuff gets into and out of blood. So it's really important to understand transport, how things are moving into cells and how things are moving out of cells. We're going to look at two types of transport today. We're going to look at passive transport and we're going to look at active transport. So let's start with passive transport first. Passive transport actually has three types within the body. So within your body, you can have simple diffusion, you can have facilitated diffusion, or you can have osmosis. So they're slightly similar with subtle differences. Let's start with simple diffusion first. It's the easiest to understand. Here, all that's happening is particles are moving from an area of high concentration to an area of low concentration. So you have a high concentration of particles here. You have a low concentration of particles here. So the idea is that particles are going to be flowing across this membrane. This actually looks just like a cell membrane. It's got a phospholipid bilayer. So particles are moving across this membrane. Since there's more particles on this side though than there is on this side, it means that the particles are going to end up moving from an area of high concentration to low concentration. So eventually these sides are going to be even. Think about food dye. If you have a drop of food dye and you put it into water, that very, very, very concentrated drop is eventually going to diffuse out and mix. And at the end of a certain amount of time, you're going to have a perfectly mixed color. That's simple diffusion. So moving from an area of high concentration to low concentration just to even out the concentrations. So simple diffusion relies on this idea of a concentration gradient. What a concentration gradient is just a difference in concentration between two areas. So you have to have an area of high concentration, you have to have an area of low concentration, and then diffusion is going to be moving from that area of high down to moving to low. If you don't have a concentration gradient, if concentrations are even, you're not going to physically see a difference with simple diffusion. You're still going to have particles moving across the membrane, but the concentrations are not going to change. Simple diffusion doesn't require any energy, and when it ends, it's called dynamic equilibrium. So dynamic equilibrium, we talk about this more actually in grade 12 chemistry. Dynamic means constantly moving, so you still have particles that are going to be moving across your membrane, but since the concentrations are equal and they're not going to be changing, we call it equilibrium. So when these concentrations are equal, this particle might move here at the exact same time that this particle moves here, but that doesn't change the overall concentration. So even though they're moving, the overall concentration is still going to be the same. Simple diffusion, for it to work, membranes have to be selectively permeable. So we talked a bit about the cell membrane before and how it lets some stuff in, but not other stuff. That means selectively permeable. So only certain substances are able to pass through by diffusion. Some of those substances that can pass through, which is really good for our respiratory and digestive system, oxygen, carbon dioxide, water, they can all pass through. So when water is being absorbed in your large intestine, it passes right through using simple diffusion. When oxygen and carbon dioxide are being exchanged at the lungs, at the alveoli, that's also using simple diffusion. Things that can't get through include ions and other large molecules. So that doesn't mean that other large molecules and ions can't pass into the cell. It just means that they can't pass into the cell using simple diffusion. So now let's talk about the next type of passive transport. This is called facilitated diffusion. So cell membranes have these things called carrier proteins that help large charged molecules get through. So a carrier protein is going to look something like this. And notice that a carrier protein is specific to whatever it's trying to let through. So this has the same shape as these circles that it's trying to let through. So a carrier protein will allow whatever that is, we call this a substrate, to bind. And then it's going to actually release that substrate into the cell. So it's going to allow certain objects to pass through it. So this can only be objects that the cell actually wants to have inside of it, because when you think about it, it's only going to have the carrier proteins shaped for things that are meant to pass into the cell. The last type of passive transport, so again, transport that doesn't require energy, is osmosis. You might have heard of osmosis before. This is the net movement of water across a selectively permeable membrane. So notice that I highlighted water. This is essentially very similar to simple diffusion, where you have water moving through a concentration gradient, but it is specific for water. So whenever we're talking about osmosis, we're talking about just the movement of water. So for example, I have a selectively permeable membrane here. I have a high concentration of water on this side, and I have a high concentration of sugar on this side. So if you take a look, I've got lots of sugar on this side, and I've got no sugar on this side, and I've got lots of water. So essentially what you want to do is, this is your high concentration, this is your low concentration. So we want to make it so there's less sugar on this side so that it's more dilute. We want to dilute the amount of sugar on this side. Now, the sugar is not able to move, but the water is able to move. So the water is going to move across the membrane. It's going to dilute the sugar, and we're going to end up with a dynamic equilibrium where the concentration of water is going to be equal, and you're going to have a dilute amount of sugar. With osmosis, you have 3 different types of solution that you can have. The first type of solution is going to be an isotonic solution. Iso essentially just means equal. So isotonic means that you have equal solute concentration. So if you take a look, this is outside the cell. This is inside the cell. You have equal amounts of solute. So we have equal amount of green substrate outside and inside. This means that there's no concentration gradient. So we're going to see no movement of water. We're going to see no osmosis. The other type of solution that you can have is a hypertonic solution. Now, what a hypertonic solution is you have an incredible amount of solute on the outside and barely any solute in the cell. So this is maybe a very, very salty solution outside of the cell. What's going to happen is water is going to want to flow out of the cell to dilute this salt. So water is going to flow out of the cell to dilute this salt. So I always remember it that hypertonic, water is exiting the cell. So in this case, water is moving out. On the other end, you can have hypotonic, which means within the cell, there's lots of solute. Outside the cell, there's barely any solute. So in this case, we want to have water dilute this solute here. So water is going to move into the cell. This is actually going to cause the cell to swell. So water moving in a hypotonic solution. How I always remember it, it's silly. I always think of O as in like hippo, like this is gonna swell up, kind of like a really big hippo. I don't know. Maybe you can come up with something better, but that's how I've always remembered it since I was in high school science. So why do we care? Who cares about water moving in the cell, water moving out of the cell? Because if your concentration gradients are off, you can actually do a lot of damage to your cells. So we have something that's called hemolysis. Hemolysis is the bursting of red blood cells. Not good. You don't want to have your red blood cells exploding everywhere. What happens is too much water is in the cells. So we actually have too much water that's moved into the cells. So in this case, you've had a hypotonic solution where a bunch of water flowed into the cells and it caused the cell to actually expand. This can be fatal. So you want to evolve not have a hemolysis scenario. You want to avoid a hemolysis scenario. That's what I was thinking about. On the other end, you can have this term called crenation. Crenation is the shrinking of red blood cells. So crenation would be like in this case here, where if you eat like a lot of salts, like a lot of salts, and you're going to have water flowing out of the cell, and the cell's going to shrink. That's an example of crenation, the shrinking of blood cells. If blood cells get too small, they're not able to function. So too little water in cells, it can also be fatal. So those are all our types of simple diffusion that we see. So in terms of transport in the cell, we've got passive transport and we've got active transport. Passive transport, which involves no energy, we looked at simple diffusion, facilitated diffusion, and osmosis. Active transport requires energy, and that is something we will deal with in grade 11 biology. So now that you understand transport, you are ready to start understanding the systems in the body.